Hi, my name is Patrick Jordan, and I'll be discussing the linkages between behavior, metabolic depression, and mortality of Unionid mussels exposed to thermal stress. Thermal stress has a significant impact on behavior and metabolism of freshwater organisms. As the climate becomes increasingly unstable, ecosystems are subjected to fluctuating temperature regimes. Mussels are especially threatened by increasing temperatures due to being ectotherms with limited mobility. We were interested in delving into the thermal tolerance of Popinaeus popii, a federally threatened species endemic to New Mexico and Texas. The first challenge we addressed in this project was that it's often hard to determine thermal limits for rare species due to limited numbers. In this case, we were only able to use 6 individuals for this study. One potential solution to this challenge would be to use a surrogate species to represent the rare species. Now, how do we decide on a proper surrogate for the species in question? One potential solution is that we try to identify members with similar behavior and physiology to the rare species. In order to do so, we need to explore what ties physiology, behavior, and thermal limits together. The first objective of our study was to compare the linkages between physiology, behavior, and mortality for the species. The next was to determine whether Popii more closely mimics the thermally tolerant or sensitive taxa. To begin, I will define the physiological endpoint. As temperature increases, respiration rate increases in response up until a certain point, after which it begins to decline. The temperature at which this occurs is referred to as MDT, or metabolic depression. Now, what does this mean? The first possibility is that it indicates an imminent onset of lethal effects due to severe stress. In this case, passing the point of MDT shows that the individual is under severe stress, suggesting that the critical thermal maximum, or CT max, would occur at just a few degrees after MDT. Or alternatively, this could mean that MDT indicates a delay of lethal effects as an adaptation to deal with severe thermal stress. In this case, MDT would serve to reduce thermal stress, ultimately reaching CT max at a higher temperature. Next, I'll define the behavioral stress indicators and mortality endpoint. Referencing the Galbraith et al. Study from 2012, we used the same behaviors as indicators of sublethal thermal stress. The first indicator is that the individuals extended the foot outside of the shell. With increasing temperature, they began to gape their valves, however, were still responsive to probing. The next 2 indicators indicated rapidly approaching CT max. At this point, the mantle tissue and siphons began to retract. And finally, they were unresponsive to probing. Provided the individuals demonstrated all 4 indicators, they were considered to have reached CT max. To select the sensitive and tolerant species, we referenced the drought tolerance study by Goff et al. In 2012. This study distinguished Pyganodon grandis as sensitive, Lampsillus straminia as a tolerant species, and these 2 species were compared against the federally threatened Popinaeus popii. These mussels were acclimated at 25 degrees Celsius for at least 2 weeks before testing, and they were fed a 2-to-1 mixture of Shellfish Diet 1800 and Nano 3600 during this acclimation period. To determine the point of metabolic depression, we used intermittent respirometry. Before the experiment, the mussels were cleaned, weighed, and fasted for 24 hours to reduce background metabolic activity. They were acclimated overnight in the chambers to limit initial stress. The individuals were then run through a repeating cycle of flush periods, which are colored in red below, where the chambers are actively exchanging water with the outside environment. The wait period, colored in yellow, is where the exchange of water is then closed off from the outside and the oxygen begins to draw down. The measure period in green is where the respiration rates were taken. This cycle was repeated throughout the experiment while the temperature was increased 2 degrees Celsius per hour. The intervals were adjusted so that the oxygen never dropped below 80% saturation. To investigate the behavioral indicators and CT max of the individuals, we set up an aerated cooler that was run simultaneously to the respirometry system. They were cleaned, weighed, and fasted to reduce that background metabolic activity, acclimated overnight to limit initial stress, and the temperature was increased 2 degrees Celsius along with the respirometry system. Now, during this trial, individuals were monitored for the 4 behavioral indicators: foot extension, valve gaping, the retraction of mantle tissue and siphons, and were unresponsive to probing. Due to respirometry being a closed system, we were unable to probe the muscles until after they were removed from the chambers. However, in the CT max cooler, subjects were much more accessible, and the 4th criterion could be verified. Now let's get into the results. On the left is the respiration rate for the thermally sensitive P. grandis. On the right, the thermally tolerant L. straminia. I'll begin by pointing out the physiological endpoint MDT. P. grandis reached MDT at 41.69 degrees Celsius. L. straminia, on the other hand, reached MDT at a lower 38.47 degrees. Now let's link the behavior to physiology. P. grandis started to demonstrate the first indicator, foot extension, at 37 degrees, about 5 degrees before reaching MDT. L. straminia, on the other hand, did not extend the foot until after reaching MDT. P. grandis began to show CT max at 43 degrees very soon after reaching MDT, with 9 out of 10 individuals demonstrating the 4 indicators. One individual did not reach CT max and was pulled alive after the experiment was terminated. For L. straminia, however, only 1 individual died at 43 degrees. 9 out of 10 individuals were still alive when the experiment was terminated at 51 degrees. To put this into perspective, 51 degrees Celsius is equivalent to about 124 degrees Fahrenheit. Interestingly, they exhibited a new behavior during this period, which was closing tightly on the extended foot. The foot then formed a curvy shape on the outside of the shell. Now, how did these 2 species compare to the model strategies we discussed earlier? According to the results, the MDT of P. grandis was an indicator that they were under severe stress and critical thermal maximum was imminent. Alternatively, the MDT of L. straminia seemed to be an adaptation to reduce thermal stress, delaying CT max until a much higher temperature. The difference between these 2 strategies could be a good gauge to determine whether a species falls in the sensitive or tolerant guilds. Now to compare with the federally threatened Poponaeus popii. They reached MDT a little under 40 degrees, with foot extension occurring at 37 before reaching MDT. They demonstrated 3 out of 4 CT max indicators at 43 degrees. as we only had enough popii to run in respirometry, we couldn't probe for response until after removing them from the chambers. So when they demonstrated the first 3 indicators, we pulled them to confirm the 4th. Now, which is Popinaeus popii more similar to? Well, popii began to show the same order of stress indicators as P. grandis, extended foot first before reaching MDT, with CT max following soon after. So to conclude, the sensitive species P. grandis demonstrated foot extension before reaching MDT, gaping soon after. The tolerant species L. straminia demonstrated foot extension after reaching MDT and did not gape. They closed tightly on the foot until the experiment was terminated at 51 degrees Celsius. So Popinaeus popii is more similar to sensitive taxa in this regard, or P. grandis in this case. Now, additional research is needed to determine if these patterns are consistent between tolerant and sensitive taxa. If so, linkages between behavioral and physiological endpoints can be used to further refine placement of mussels into gills, the importance of which has been described in previous papers such as Gates et al. In 2015. I would like to thank CEHMM, U.S. Fish and Wildlife, and Auburn University SFAAS for helping me out with mussel collections and funding. And I'd like to thank Evelyn Pieper and Gabby Elliott for helping me out in the field. Thanks so much for listening.